to repair. Okay? So of course, people who are actively growing need more nutrients. Their caloric intake is increased. People who are recovering from like burns or accidents need more nutrition. The major nutrients, of course, are carbohydrates, lipids, and proteins. And you learned about these in A&P 1. So some of what we're going to go over, you already know. We'll talk about them in A&P 1. Now, other nutrients, minor nutrients, include vitamins, minerals, and water. Okay? These nutrients that are taken in our diet undergo metabolic reactions, both inside the GI tract and within the body. You know that within the GI tract, the type of metabolic reaction is one in which we take a larger molecule and break it down into its building blocks so that it's small enough to be absorbed. For instance, you know, we break carbohydrates down into monosaccharides, we break proteins down into amino acids, and we break lipids down into fatty acids. Well, those types of reactions that occur within the digestive tract where we take larger molecules and break them into smaller ones, they're referred to as catabolic reactions. Catabolic reactions take big and create small. On the other hand, some of the reactions that occur within the body following the absorption of those building blocks are anabolic reactions, where we take then the building blocks and produce larger molecules. For instance, those amino acids, once they reach the bloodstream, they may be picked up by muscle tissue and converted into muscle proteins, like actin or myosin. So, when we talk about metabolism, that metabolism represents the sum total of all the chemical reactions that occur within the body. Okay? And some of these reactions are anabolic, some are catabolic, but they're all metabolic. Let's talk about carbohydrates. Carbohydrates, I think you know from A&P 1, have this chemical formula. C, N just stands for some type of number, H2O, with another number behind it. So they're hydrates, H2O, of carbon, carbohydrates. Okay? Most frequently, the N and the P are the same number. In fact, for the simplest type of carbohydrate, like glucose, this number is often 6. So the molecular formula of glucose would be C6, and then if the P is also a 6, we multiply that times the 2, so it's H12O6. In other words, there's a 1 to 2 to 1 ratio between carbon and hydrogen and oxygen. 1 to 2 to 1. Again, the simplest carbohydrates are called monosaccharides. And typically within the body, these monosaccharides are composed of a backbone of either 5 or 6 carbons. So nutrients like glucose and fructose and galactose, all of these have 6 carbons. So all have the same molecular formula, C6H12O6. It's just that the atoms are arranged around the ring in a little bit different order. Okay? So the monosaccharides that are most common in our diet, that are derived from our diet, are going to be glucose. That's the principal sugar used as a fuel for our tissues. Fructose, that's a type of sugar found in fruit juices and honey. That's fructose. And galactose is found in milk. When I say sugar, I'm going to use that term interchangeably with monosaccharides. Now, disaccharides are simply 2 monosaccharides linked together by a covalent bond. Within the GI tract, if a disaccharide undergoes hydrolysis, remember hydrolysis means to cleave with water, then it's going to yield 2 monosaccharides, which can then be absorbed. Now, the disaccharides you need to know are sucrose, lactose, and mannose. Sucrose is composed of glucose and fructose. Lactose is glucose and galactose. And mannose is two glucoses bound together. Okay. Remember that for each of these key disaccharides, there is a disaccharide present along the brush border to break it down. Okay. So for sucrose, that disaccharide is going to be called what? Sucrase. For lactose, it's going to be lactase. And for mannose, it's going to be mannose. And that's going to be found along the brush border. Those are produced by the small intestine. Okay? Now, if I take a bunch of monosaccharides and link them together into a longer chain, then I have what's called a polysaccharide. Polysaccharides are larger polymers which yield 8 monosaccharides through hydrolytic reactions. Okay. So, for instance, starch. It's a storage polysaccharide. It's made up of glucose building blocks. But this is the way that plants store their glucose, their excess glucose. It would be analogous to glycogen in animals. Okay? So in fact, most of the carbs that are ingested in our diet come in the form of starch, okay, which is just a big old polysaccharide made up of glucose. It's produced by plants. Okay. Remember that within our GI tract, when we digest the starch, it's going to be broken down. Where does that carbohydrate digestion begin? In the mouth, yeah, through the activity of salivary amylase. Okay, now that salivary amylase is inactivated once it reaches the stomach. So digestion continues then in the small intestine through the activity of what enzyme?
Pancreatic amylase.
Pancreatic amylase. And the pancreatic amylase is going to cleave off sugars from that larger molecule, leaving you mostly with monosaccharides and disaccharides. Those disaccharides can then be broken down by brush border enzymes, those disaccharide aces we talked about. Well, in the same way, proteins from our diet are going to be broken down into amino acids, and fats are going to be broken down into glycerol and fatty acids. That's what happens within our GI tract. Stuff is broken down so that the smaller building blocks are small enough that they can be absorbed across the lining of those cells that make up the epithelium of the GI tract. Of course, in the small intestine, that would be a simple columnar epithelium. Just got to put it in there. Yeah, good that way. So now, as a result of carbohydrate digestion, we produce molecules of glucose and fructose and galactose. And all of these will be absorbed across the small intestinal mucosa, and they'll enter into the superior mesenteric vein, which carries them to the portal vein, which then carries them to the liver. And the liver is the first crack at these molecules. Now, what the liver will do is it can utilize glucose as a fuel, so can other cells throughout the body. But if in fact the monosaccharide is fructose or galactose, what it will do first is convert those to glucose, so that then they too can be utilized as fuel. Now, it's interesting that I say that glucose is the main fuel used by most tissues. The fact is that tissues have the ability to utilize other substrates as fuel as well. For instance, most tissues, most cells can utilize fat as a source of fuel. And in fact, for certain tissues, that's the preferred source of fuel. But there are two types of cells, those that make up the central nervous system, the brain, neurons, and red blood cells that rely almost exclusively on glucose. So red blood cells and the brain, they can't necessarily use fat. They rely on glucose. And so, and neurons are very metabolically active. there's a constant demand for glucose within the brain. So that if you become hypoglycemic, okay, that leads almost immediately to central nervous system dysfunction. So if you become severely hypoglycemic, you may become confused, disoriented. If it's severe enough or prolonged enough, you may become obtunded or comatose. Why? Because the brain has a constant demand for glucose. All right. Now, when glucose is taken into cells, its initial breakdown, its initial catabolism, yeah, I said it, catabolism, occurs within the cytoplasm. Okay? Through an anaerobic pathway, anaerobic meaning it doesn't require oxygen. That anaerobic pathway is called glycolysis. And all of the enzymes that are necessary to carry out this pathway, this series of metabolic reactions, are present within the cytoplasm. So glycolysis does not require oxygen. It occurs in the cytoplasm. You know, if you take biochemistry, you have to learn every step of the pathway. And when you take glucose and then you give it a phosphate handle, or glucose 6-phosphate, and then you add another phosphate handle, and then you break it up. You know, you split it. And so it's like, it might be like 10 steps, each step of which is catalyzed by an enzyme. But as a result of the breakdown and rearrangement of glucose, the end product is 2 molecules of pyruvate. See, pyruvate is a 3-carbon molecule, so you preserve the number of carbons. It's 6 carbons. Now, when you break bonds, you release energy. When you rearrange molecules, okay, and form bonds, most of the time you utilize energy. So the net effect of this chemical pathway is you generate a net of 2 molecules of ATP. Okay? So glycolysis can yield energy from that glucose. But it's not a very efficient method of generating ATP. If oxygen is present, we can pull a lot more ATP from this original glucose molecule. What we do is we take the pyruvate and then we shun it into the mitochondria. That's kind of a cigar-shaped organelle. It looks kind of like this. Okay? And along the inner cell membrane of this mitochondria, you have enzymes that catalyze the steps in a different metabolic pathway called the Krebs cycle. It's also called the citric acid cycle. Okay? But this series of reactions that occurs within the mitochondria requires oxygen. So it's an aerobic pathway. But in the presence of oxygen, then we can extract more ATP. So pyruvate gets shunned into the mitochondria, where it's converted into a different molecule called acetyl coenzyme A. This acetyl coenzyme A is the starting point of the Krebs cycle. It's a 2-carbon molecule. And in the Krebs cycle, it's going to be joined by a 4-carbon molecule. And then that intermediate is going to undergo another reaction, then another reaction, then another reaction. Okay? It's a cycle. Until then, finally, at the end, we generate some more acetyl coenzyme A. This Krebs cycle, a series of reactions requiring oxygen that occurs within the mitochondria, is much more productive than glycolysis. This cycle generates about 36 molecules of ATP. Okay? So that as a result of the breakdown of glucose, we generate 36, an average of about 36 molecules of ATP. And someone might say, well, what about that other 2 here? That would be 38. Well, it takes some energy to move pyruvate into the mitochondria. So it ends up being somewhere closer to 36.
So each pyruvate makes 36?
No, the total, the 2 when you enter here. So from 1 glucose, you get 36. Okay. Now, from that same glucose molecule, as a result of this series of reactions, you generate 6 molecules of CO2 and 6 molecules of water. So when cells are breaking down glucose, they're generating CO2. And of course, that CO2 has to be disposed of. So that CO2 is going to leave the tissue, enter into the capillary blood, and it's going to be returned to the lungs where we then exhale it. Within the lungs it undergoes external respiration. It crosses that respiratory membrane into the alveoli, and then we simply blow it off. Did everyone follow that? Because in my head I have it straight, but if I were hearing it for the first time, I'm not sure I would, necessarily pick it up that quickly. So if you have a question, just raise your little hand. Did you follow?
It's like,
I think you talked about it before. If you want.
Well, that's keto. The truth is, only a small amount which you take in AP1 is retained. You can spit it right back out. It's down there somewhere. When you take another course and there's some overlap, it brings it back to the surface. It's more likely to become part of long-term memory. And so as you keep taking courses, that happens repeatedly. You overlap and you refresh your memory. You freshen it up. And it brings it to the surface. And it becomes more part of long-term memory. Okay. So this just demonstrates the glycolytic pathway where we take glucose and form 2 molecules of pyruvate. And on the other hand, this line discusses the Krebs cycle. You see, this is like if you were to take biochemistry, like you would have to learn each of these steps, each of the intermediates, each of the enzymes that catalyzes it. But if you take biology, this is what it looks like. It's a much simpler version. Now, what happens, though, if you don't have sufficient oxygen or sufficient oxygen delivery to the tissue? Let's suppose you go into shock. Your blood pressure drops. And blood pressure is what drives blood flow. And so, in light of the drop in blood pressure, you can't deliver enough blood flow with oxygen to tissues. Well, in that case,